Okay, so I want you to picture a scene. And it's a scene, we have all lived 100 times. You're at the beach,
Right, the classic summer day.
or maybe you're just out gardening in the backyard or sitting on a nice patio having lunch. The sun is beating down. It feels glorious.
It absolutely does. It's the feeling we wait all winter for.
Exactly. And because you are a responsible, well-informed person,
Mm-hmm.
because you listen to deep dives like this one, you've done the prep work. You've got the hat.
Check.
You've got the sunglasses.
We check.
And crucially, you have slathered yourself in SPF 50.
Right.
You're blocking those UV rays. You're stopping the burn. You're stopping the DNA damage. You feel safe. You feel protected.
And, I mean, biologically speaking, you are safer. We have spent the last 40 years at least drilling the dangers of ultraviolet radiation into the public consciousness. Yeah. UVB burns you, UVA ages you. That's like the fundamental mantra of modern dermatology.
Well, that's the baseline. We all know the rules of that game. But here is the uncomfortable question I have for you today. What if I told you that while you're successfully blocking the light, you are completely ignoring the heat?
That is the missing variable. It's the ghost in the machine for almost everyone's skincare routine.
I'm not talking about the light spectrum here, not UV, not visible light. I'm talking about pure thermal energy, infrared radiation. What if I told you that the heat itself, just the sheer rise in temperature of your skin, is aging you just as fast, if not, faster than the UV rays you're so worried about?
It's a concept that dermatology is really, I'd say, just beginning to take seriously in the mainstream. We call it thermal aging.
Thermal aging.
And it's insidious because unlike a sunburn, which you see immediately, you turn red, you peel, you know it's there. Thermal aging happens silently. It's deep in the dermal layers. You don't see it until years later when it's too late.
Thermal aging. It sounds so industrial. It sounds like something that happens to a car engine or like a power plant turbine. It doesn't sound like something happening to my face.
In a way, it is similar. It's wear and tear. It's damage caused by an energy overload. But here's the kicker. It's not just the sun.
Right. See, this was the part of the research stack that actually panicked me a little bit.
I get it.
Because I can avoid the sun, I can wear a hat, I can stay in the shade, but I cannot avoid, my life.
Exactly. I mean, think about your daily routine. You blow dry your hair in the morning.
Every single day.
That is a thermal blast directed right at your scalp and your face. You open a hot oven to check on dinner.
That wave of heat that hits you.
A thermal blast. You sit in a sauna because you think it's good for you. It's detoxifying.
Also a thermal blast.
It is, and the one that affects almost everyone, the one that I am definitely guilty of probably 10 times a day, holding a smartphone against your ear for a 40-minute conference call.
Ugh, that one hit home for me too. You finish the call, you pull the phone away, and your ear and your cheek are physically hot to the touch. You can feel the warmth just radiating off the glass.
You're creating a localized heat pocket.
A heat pocket?
Exactly. And inside that little pocket, your skin cells are panicking. They're triggering inflammation. They're breaking down their own collagen, all because of that specific thermal stress. It is a silent, accelerated aging process happening every single time you make a call.
So we're essentially cooking ourselves.
Yeah.
Slowly. Like a sous vide.
That is a disturbingly accurate analogy.
And this is why I love doing these deep dives. We aren't just here to deliver bad news and make everyone paranoid about their hair dryers.
No, and that's not the mission.
We found a solution in the research. And it is completely unexpected.
To say the least. It's a left turn I really didn't see coming when I first opened the source material.
The solution to this high-tech, modern problem of thermal aging from our smartphones isn't found in some sterile lab in Silicon Valley. It isn't some synthetic polymer cooked up by chemists.
Nope.
It's found in the dirt, specifically under a spruce tree in the Rocky Mountains.
It really is a fascinating pivot. We're going from discussing, high-tech infrared radiation and the thermodynamics of a smartphone to talking about a mushroom, a very specific and, I have to say, somewhat ugly mushroom called Albatrellus ovinus.
The Sheep Polypore, which I think is just a delightful name.
It is a fitting name, as we'll see.
So today we are going on a mission. We are going to figure out how this humble terrestrial fungus holds a chemical key, something called Grifolin, that can literally reach into your skin cells and flip the off switch on your pain and heat receptors.
We're going to bridge the gap between classic mycology, the study of fungi, and very advanced molecular biology. Our stack of sources today really ranges from identification guides by the Wisconsin Mycological Society all the way to some heavy-hitting pharmaceutical reviews from the World Journal of Pharmaceutical Research.
It is a bit of a whiplash mix. We've got how to spot a mushroom in the woods on one side,
Right.
and TRPV1 antagonist receptor mechanics on the other.
But that's where the magic happens. It's always in that intersection. That's where real innovation comes from.
So buckle up. We're going to learn how to identify this mushroom, we're going to learn how to trick our skin into ignoring heat, and we're going to build what the sources call a botanical arsenal to keep our collagen intact. Let's dive in.
Let's do it.
Okay, let's start with the hero of our story, Albatrellus ovinus, the sheep polypore. Now, I have to be honest with you. When I hear hero mushroom or, medicinal mushroom, I picture something striking.
Sure.
A bright red reishi, or a glowing lion's mane, or maybe those bright orange cordyceps you see in documentaries.
Of course, the charismatic megafungi, the ones that end up on the cover of magazines.
Exactly. But I looked at the photos from Michael Kuh's Mushroom Expert site, which is our go-to for identification, and this guy is humble? Is that the polite word for it?
Humble is very polite. Drab is probably more accurate for most of its life. If you were walking through a conifer forest in the Pacific Northwest, or the Appalachians, or maybe Northern Europe, you would probably step right on it without even noticing. It really does blend in.
So paint the picture for us. What are we looking for if we're out hiking?
Okay, so first off, it's a polypore. Now, usually when listeners hear polypore, they probably think of those hard, woody shelves growing out of the side of a dying tree.
Like turkey tails or artist's conch. The ones you can practically stand on.
The ones that would absolutely break your teeth if you tried to take a bite out of them.
Right.
Exactly. Those are wood decay fungi. But the whole genus Albatrellus is distinct because it is terrestrial. It grows on the ground, rising right out of the soil, just like a standard cap and stem mushroom. It looks like a normal mushroom, for lack of a better word.
So it looks like a toadstool, but it acts like a polypore.
Structurally, yes. If you were to pick it up and flip it over to look under the cap, you won't see gills. You won't see those radiating fins like you see on a portobello or a button mushroom.
Okay.
Instead, you'd see a surface that looks kind of flat and white at first glance, but if you look really closely or maybe use a hand lens, you'll see it is just covered in thousands of tiny microscopic holes. Pores.
Hence polypore. Many pores. Okay, so cap on top, tiny holes on the bottom growing out of the dirt. What color is it? Is it vibrant at all?
Well, that kind of depends on its mood, or maybe accurately its age. When it is young and fresh, it can be this sort of pristine, creamy, white. It looks quite clean, almost marshmallow-like in texture.
Oh, that sounds nice.
It does, and Kuo is very funny about this in his description, it does not stay that way for long. As it ages, it turns into this kind of tan, brownish, leathery colored thing.
And it cracks. The pictures show these deep cracks.
It cracks significantly. The surface of the cap just breaks apart like a dry desert floor. You get these tessellated cracks that reveal the white flesh underneath. It looks weathered. It looks like it's had a hard life.
I have to say, I felt a deep spiritual connection to the description. Pristine when young, drab and cracked when older. I thought, same, mushroom, same.
It happens to the best of us. But that cracking is actually a really helpful identification feature for foragers. Because you have to remember, if you're in the woods looking for this, you're usually looking down at a carpet of brown pine needles.
Right.
You need those distinctive features to distinguish it from just a rock or a pine cone or another LVM, a little brown mushroom.
And you're not finding these under oak trees or maples, are you? The notes were really specific about the habitat.
Very specific, and this is a crucial concept to understand. Albatrellus ovinus is mycorrhizal.
Mycorrhizal.
It's not just eating dead leaves on the ground, which is what a saprobic fungus would do. It is living in a symbiotic partnership with the roots of living trees.
A partnership.
A partnership, specifically with conifers. Engelmann spruce, subalpine fir. The sources are very clear on this.
So it's actually trading nutrients with the trees.
That's exactly it. The mushroom's mycelium, its underground network, is far better at pulling certain minerals and water from the soil than the tree's roots are.
Okay.
So the mushroom gives the tree those hard-to-get nutrients, and in return, the tree, which can photosynthesize, gives the mushroom sugar. It gives it carbon. It's a trade agreement. So if you aren't seeing spruce or fir trees, you are not seeing the sheep polypore, period.
Okay, so I'm in the Rockies. I see a spruce tree. I look down. I see a drab, cracked, tan mushroom. And it's probably growing in a cluster, right? Because they like to huddle together like sheep, hence the name.
That was one of the theories on the name Ovinus, that it relates to sheep or that it grows in these flocculent clumps. The other theory is that sheep actually like to eat them.
Oh, interesting.
But yes, they often grow in these dense fused clusters where the stems actually sort of melt together at the base into a single mass.
So I think I've found it. But as with all things in nature, it's tricky. There's a doppelganger.
There is always a doppelganger in mycology. It's rule number 1. And in this case, it's the quote-unquote evil twin, Albatrellus confluens.
Confluens. Even the name sounds a bit slippery, a bit suspicious.
And it's a classic forager's dilemma because confluens grows in the exact same spots. It looks almost identical. It's tan. It's a terrestrial polypore. It grows in clusters. Visually, to the untrained eye, they are twins.
So how do we know we have the magical pain-killing mushroom and not the bitter imposter? Because confluens tastes bad, right? That's the main issue.
It's quite bitter, yeah, and generally considered inedible. It's not dangerously toxic. It's not going to kill you, but it will absolutely ruin your dinner.
Right. So we want to avoid that. How do we tell them apart? I love this part because it involves bringing a little chemistry set into the woods with you. It feels like CSI. Forest edition.
It really does. It requires the KOH test, potassium hydroxide. And serious mycologists, they actually carry a little dropper bottle of this stuff in their pockets when they hike. It's a strong base.
It's basically liquid drain cleaner, isn't it? Just a little bit.
A diluted form, of course, but yes, it is caustic.
Yeah.
And it serves as a kind of chemical litmus test for fungal flesh. So picture this. You're on the forest floor. You have this mystery mushroom. You slice a tiny piece of the cap. You take your dropper and you put a single drop of KOH on the white flesh inside.
And if it's our hero, if it's the Sheep Polypore, Albatrellus ovinus,
it instantly turns a vivid, dirty, golden yellow.
Golden yellow. Like it's revealing its true value. It's showing you the gold inside.
That's very poetic, yes.
Shit.
But if it is the imposter, if it is Albatrellus confluens...
Don't tell me. It melts. Or turns black.
It turns purple.
Purple. That is such a distinct difference. There's no ambiguity there. Yellow for yes, purple for pass.
It is remarkably reliable. It's one of those great party tricks of mycology. And that distinction really matters, not just for dinner, but for our whole conversation. Because Albatrellus ovinus has a long history of being consumed.
Right.
In Finland, the Wisconsin Mycological Society notes it's sold in markets. It's considered a fine edible.
So what does a fine edible taste like? Does it taste like medicine? Because usually things that are good for your skin taste like, dirt.
Not at all, apparently. It's described as being very mild with a distinct almond-like flavor.
Almonds. Okay. That is surprisingly pleasant. I was expecting something bitter or earthy.
Which is actually really important for our discussion today, because we are about to talk about extracting some very potent chemicals from this thing and rubbing them all over our faces.
Right. It implies a safety profile from the get-go.
Exactly. If this was some toxic mushroom that happened to block pain receptors, like, say, the way some snake venoms block nerve signals, we would have a much harder time bringing it to the skincare market.
For sure.
We would have to worry about side effects, toxicity thresholds, all of that. But because it has this centuries-long history of being eaten without ill effect, we start from a place of safety. We start with generally recognized as safe.
We know it doesn't kill you. It just tastes like almonds and turns yellow when you put chemicals on it.
A great starting point for any new skincare ingredient.
So we've identified the mushroom. We know where it lives. We know it's safe. But how in the world do we get from a forest in Finland to a high-tech anti-aging cream? What is hiding inside that almond-flavored flesh that researchers found so interesting?
This is where we leave the woods and we enter the laboratory. And the researchers, specifically at a Swiss cosmetics biochemistry firm called Rahn AG, they took this mushroom and they performed an extraction. They used a specific solvent called 1,4,3-propanidyl.
Okay, why that specific solvent? Why not just use water or alcohol?
Because the compounds they were looking for, the active ingredients, they aren't water-soluble. They are what we call lipophilic.
Lipophilic, or fat-loving.
Fat-loving, exactly. So 1,4,3-propanidyl is really good at pulling those specific types of molecules out of the fungal cell walls without damaging them.
And what did they find when they analyzed this extract? What was the goal they were mining for?
They found a very specific class of molecules called prenylated phenols.
Prenylated phenols. That is a mouthful. Try saying that 5 times fast. What does that mean in English?
So let's break it down. A phenol is a very common chemical structure. It's a ring that you find all over nature. You find phenols in wine, in green tea, berries. They're often antioxidants.
Okay, I'm with you.
But the prenulated part is the key twist here. That's the special sauce.
What is a prenyl group?
A prenyl group is a little hydrocarbon chain that's attached to that main phenyl ring. And you can think of it as a grease anchor.
A grease anchor, I like that.
Yeah. It makes the whole molecule much more slippery to fats. It allows the molecule to slide easily into and through cell membranes, which, remember, are made of lipids. They're made of fats. Without that prenyl anchor, the phenyl molecule might just bounce off the cell. With the anchor, it can slide right in and get to work on the inside.
So it's like a VIP pass. It's a key that gets it through the cell's bouncer at the door.
That's a perfect way to put it. And the specific molecules they found in the Sheep Polypore are named Grifolin, neogrifolin, and scutyrol.
Grifolin. Sounds like a wizard from Lord of the Rings. Grifolin the White.
It practically is, because Grifolin has the power to cast a silent spell on your nerves.
I love that image. So we've got this molecule, Grifolin, with its little grease anchor, and it's sliding into our skin cells. What is it looking for? What is its target once it's inside?
It is hunting for a very specific protein, a gatekeeper on the cell surface called TRPV1.
And this is the villain of our story, TRPV1.
Not really a villain, more like an overzealous, paranoid security guard. But to understand why we need to shut him up, we first need to understand what he's shouting about all the time.
Okay, let's unpack this. TRPV1, that stands for Transient Receptor Potential Vanilloid 1.
It is a bit of an alphabet soup, I know.
I know vanilloid makes me think of vanilla, but I assume this has nothing to do with ice cream.
Ironically, no. It actually refers to the chemical group that includes capsaicin.
Capsaicin, the stuff that makes chili peppers hot.
The very same.
Okay, so this receptor, this TRPV1, it's sitting on the surface of our nerve cells.
Yes, on sensory nerve fibers, but also, and this is absolutely key for our discussion, it's found on keratinocytes. Those are your primary skin cells, the bricks that make up your epidermis. So imagine this receptor as a tightly closed gate sitting in the cell wall. It's just waiting.
Waiting for what?
It's waiting for noxious stimuli, for danger signals. It's an ancient system that's evolutionarily designed to stop you from hurting yourself. It triggers and flies open when it senses high heat, specifically temperatures above about 42 or 43 degrees Celsius.
That's hot. That is a very hot bath. That's the ouch point.
It is the ouch threshold, exactly. It also triggers in response to acid, so low pH. And most famously, as we said, it triggers in response to capsaicin.
So when I eat a ghost pepper wing, and I feel like my mouth is literally on fire.
Yeah.
That's not actual heat. My tongue isn't burning.
No, there is no thermal damage happening whatsoever. You are not being burned. But the capsaicin molecule has the perfect shape to fit into the TRPV1 receptor like a key. It twists the lock and forces that gate to fly open.
And once the gate is open, what happens then?
Calcium ions, which are waiting outside the cell, rush in. That sudden influx of calcium is the electric signal. It screams up your nervous system to your brain, and it says one thing: fire, burn, pain.
So the brain doesn't actually know the difference between a chili pepper and a hot stove.
Not really. To the brain, the signal is identical. Get away from this thing. It is causing damage.
That seems like a flaw in the system.
Right?
Why do we have a receptor that gets hacked by a vegetable?
Well, from the plant's perspective, it's not a flaw. The vegetable evolved that chemical specifically to stop mammals from eating its fruit and destroying its seeds. It's brilliant biological warfare, but usually TRPV1 is a good system. You want to know if you are touching a hot stove.
True.
You want to pull your hand away before your skin melts.
True. I would like to keep my skin from melting. That's a life goal.
Yeah.
But we're talking about skincare here. We're talking about so-called sensitive skin.
Right. And this is where it gets really interesting for all the listeners out there who identify as having sensitive skin. The research strongly suggests that people with sensitive skin might have a structural issue with their TRPV1 receptors.
What kind of issue?
It could be one of two things. Either they have too many of them, their skin is like a wall with too many windows, or the receptors they do have are just hair trigger sensitive. They open too easily.
So the security guard is paranoid.
Extremely paranoid. A warm breeze, alarm, a slightly spicy meal the night before, alarm, a new face cream with even a tiny bit of an active ingredient, full-blown 5-alarm fire.
And that alarm on the skin looks like redness, stinging, itching.
All of the above. We call it neurogenic inflammation. The nerves themselves are releasing inflammatory chemicals because they think they're under attack, even when they're not.
So conventionally, how have we dealt with this? If I have sensitive skin, usually I'm just told to.
Yeah.
Avoid everything.
Yeah.
Use bland products. Don't look at anything spicy.
Avoidance is one strategy for sure. The other is something called desensitization. Which brings us back to people who love spicy food.
They build up a tolerance.
Right. If you hammer that TRPV1 receptor with capsaicin over and over and over again, eventually it gets exhausted. It actually retreats from the cell surface. It stops firing. It is like shouting at the security guard until he goes deaf and walks off the job.
So the strategy is hurt it until it stops hurting.
Basically, yes.
Right.
High-dose capsaicin patches are actually an FDA-approved treatment for neuropathic pain, but you have to endure the intense burning first to get the relief later.
And for a cosmetic product. For an anti-aging cream you buy at the store.
It's a complete disaster. You cannot sell a face cream that burns like fire for 20 minutes and tell the customer, don't worry, it'll feel great next week.
No, that is a 1-star review immediately. I am returning that so fast.
Of course. So we need a different mechanism. We don't want to overstimulate the receptor to death. We want to block it. We want to put a mute button on it. We want to stop it from firing in the first place.
We want to jam the lock so the capsaicin key doesn't even fit.
Precisely. We need a TRPV1 antagonist.
And bringing it all the way back to our mushroom, that is what Grifolin does.
That is exactly what Grifolin does. It acts as a non-competitive antagonist. It binds to the receptor at a different site and physically prevents the gate from opening. No gate opening, no calcium rush, no pain signal to the brain.
And no inflammation.
And no subsequent inflammation.
You know, there was a technical term in the source material about this binding, the Hill coefficient. It sounded very math-heavy, and honestly, my eyes kind of glazed over. But you seemed excited about it in the pre-show notes. What is that?
I was, because the Hill coefficient tells us how it binds. It's a measure of something called cooperativity.
Cooperativity, like teamwork.
Exactly like teamwork. So the TRPV1 receptor is actually a tetramer. It has 4 identical parts that work together to form the channel. A Hill coefficient of 1 means one molecule binds to one spot, end of story. It's a 1-to-1 interaction. But the study showed Grifolin has a Hill coefficient of roughly 1.5 to 2.4.
Which implies?
It implies that when one molecule of Grifolin binds to one of the 4 parts, it makes it easier for the next molecule of Grifolin to bind to another part. They recruit each other, they help each other out.
Oh, that's cool. It's like a tug of war.
It's exactly like a tug of war. Imagine trying to hold a big, heavy double door shut while the wind is blowing it open. If you do it alone, it's really hard. But if you grab one handle and that somehow signals your friend to immediately grab the other handle, suddenly the door is much more secure.
So it's not just jamming the lock. It's like a whole squad of Grifolin molecules linking arms and holding the door shut together against the heat.
That is a great way to visualize it. It creates a very stable, very effective block on that receptor.
Okay, that's the theory. In a petri dish, Grifolin blocks the receptor.
Oh.
But does it work on actual people? Because my skin is definitely not a petri dish. Usually, things that work great in the lab fail spectacularly in the real world.
This is where the Rahn AG study gets really fun. They did an in vivo study on living humans, and they recruited a very specific group of people. They called them stimmers.
Stingers. That sounds like a sci-fi faction. Or a very bad high school sports team.
It's a technical term in cosmetology, actually. These are people who are known to have a strong, stinging reaction to things like capsaicin or lactic acid. They have scientifically verified sensitive skin.
Got it. So what did they do to the stingers?
The researchers applied a cream containing 3% Albatrellus Ovinus extract to one side of their face, specifically on the nasolabial fold.
The laugh lines. That's a very sensitive spot on the face.
It's very sensitive. And then they applied a placebo cream, a cream with no mushroom extract, to the other side, a classic split-face study.
And then they pepper spray them.
Not quite pepper spray, but close. They applied a standardized capsaicin solution to both sides to trigger that pain and stinging response.
What was the
The results were significant and immediate. The side of the face with the mushroom extract had much less stinging, much less burning, and visibly less redness. The study used a laser Doppler to measure microcirculation.
Which is just a fancy way of saying blood flow to the surface.
Exactly. And the blood flow was significantly reduced on the mushroom side.
So the mushroom extract basically told the skin, hey, chill out. It's just a little chili pepper. Don't freak out. No need to call in the inflammatory calorie.
Precisely. It silenced the alarm. But remember, our main topic isn't just about avoiding the sting of hot sauce on your face. It's about thermal aging.
Right. The heat itself.
So they did another test, a heat threshold test. For this, they used a device called a thermode.
A thermode?
It's basically a highly precise heating pad. Think of it like a scientific soldering iron with very, very accurate temperature control. They placed it on the subject's forearm, and slowly, steadily ramped up the temperature. And they asked the subjects to signal the exact moment they felt pain.
To find their unique pain threshold.
Yeah, right. For most people, that's usually around 44 or 45 degrees Celsius. So they established a baseline, then they had the subjects use the mushroom cream on that arm for 14 days, and then they did the test again.
And what happened after 2 weeks?
Their pain threshold went up by 1.5 degrees Celsius.
1.5 degrees. That sounds small. I mean, if I turn my thermostat up 1.5 degrees, I can barely notice the difference.
In environmental terms, it seems small. But in biology, in physiology, that is a huge buffer. Think about a fever. The difference between a normal body temp of 37 degrees and a fever of 38.5 degrees is massive. You feel awful. Your whole body's biochemistry has changed.
That's a really good point. Okay.
So increasing the skin's heat tolerance by that much means your skin can endure significantly more thermal energy before it triggers that inflammatory cascade we've been talking about. It means you have fundamentally increased the skin's resilience to its environment.
So we've established that the sheep polypore can numb the skin to heat and pain. That's amazing. But I want to circle back to the why. Why does heat even matter for aging? I get that UV rays destroy DNA. I get that smoking destroys collagen. But heat? Is it really just cooking us like a slow roast?
It's surprisingly similar to UV damage, but the pathway is a little different. And this is where we need to get into the biochemistry of destruction.
Lay it on me. I'm ready.
Okay, so heat hits the skin. Whether it's from the sun's infrared rays or from your phone, that thermal energy excites the cells. Specifically, it puts a lot of stress on the mitochondria, the little power plants inside every cell.
And when mitochondria get stressed out, they get leaky. They make mistakes.
Exactly. They start pumping out ROS-reactive oxygen species.
Free radicals, the bad guys of every skincare commercial.
The ultimate vandals of the cellular world. Now, usually your body has antioxidant systems to clean these up, but if there's too much heat for too long, you get an overload of ROS. These free radicals then trigger a signaling pathway. They flick a switch that activates a group of proteins called MAPKs.
Mitogen-activated protein kinases. I vaguely remember this from biology class.
Think of MAPKs as the panic-stricken messengers. They run from the site of the damage to the nucleus of the cell, the command center, and they deliver an urgent message. And that message is, we are under attack. Initiate self-destruct. Destroy the structure.
Why? Why would the cell want to destroy itself? That seems counterintuitive.
It's a scorched-earth policy. It's trying to clear out damaged tissue to make way for rebuilding. But under conditions of chronic heat exposure, it just keeps destroying and destroying. So the nucleus responds to the MAPK message by producing a group of enzymes called MMPs, matrix metalloproteinases.
MMPs. I always imagine these as little Pac-Men.
That is the perfect image. MMPs are enzymes that are designed to chew things up. Specifically, they chew up your collagen and your elastin, the very proteins that give your skin its structure and bounce.
Collagen is the scaffolding that holds our skin up and keeps it plump and firm. So if the Pac-Men are released and they eat the scaffolding,
The whole structure collapses. You get wrinkles, you get sagging, you get that leathery, thick texture you see on people who have spent their whole lives in the sun. We used to think that was all UV damage. But we now know a huge chunk of that is actually thermal damage.
wow. So let me try and connect all the dots here.
Yeah.
Heat hits the skin. That triggers the overzealous TRPV1 receptor.
Yep.
Which causes immediate inflammation.
Yeah.
That inflammation creates a flood of free radicals.
The ROS, yeah.
The ROS wake up hin. The Pac-Men, the MMPs, and the Pac-Men eat my collagen.
That is the entire chain reaction from start to finish. And the Albatrellus Ovinus extract, the Grifolin, works at the very, very top of that chain. By blocking the TRPV1 receptor, it stops that initial panic signal. It calms the inflammation before it can snowball into collagen destruction.
So it's preemptive. It's not just trying to clean up the mess afterwards. It is stopping the mess from ever happening in the first place.
Exactly. But, and this is crucial, the source material, especially that big review paper, suggests that for the best possible protection, you shouldn't rely on just one mechanism. The mushroom is great, but it's just one soldier in the army. For a truly robust defense, you want what they call a botanical arsenal.
I saw that in the World Journal of Pharmaceutical Research Review. It wasn't just mushrooms. They listed this whole cocktail of herbs designed to fight thermal aging.
Right. Because while the mushroom is blocking the signal of the heat, you still have to deal with the physical effects of the heat that is already there and, the free radicals that might slip through the cracks. You need a backup plan. You need multiple layers of defense.
Okay, so let's meet the rest of the squad in this arsenal. First up on the list was something that sounded really delicious. Apple mint. Mentha suaveolens.
And this isn't your standard toothpaste peppermint or spearmint. Apple mint is a different species entirely. It has these rounded, fuzzy leaves, and it smells distinctly fruity, almost like green apples.
And I assume we aren't just using it in the cream because it smells nice.
No, though that definitely helps in a cosmetic product. The extract is made from the leaves, and it's incredibly rich in flavonoids and polyphenols.
And what is its superpower in this war against heat? What's its specific job?
Apple mint's job is to target the ROS of free radicals directly. Remember, even if we block the pain signal with the mushroom, the heat energy itself is still physically creating some of these free radicals in the cell. Apple mint is the cleanup crew.
So it's a scavenger? It mops up the free radicals.
Yes. But even more specifically, it suppresses those MAPK messengers we talked about.
The ones that run to the nucleus and scream self-destruct.
Those are the ones. So if the mushroom is the security guard stopping the initial breach at the gate, Apple mint is like a secret agent intercepting the messenger before he can deliver the orders to the command center to release the Pac-Man.
I like that. Defense in depth. A multi-layered strategy.
Exactly. And then we have the real heavy hitter from Ayurvedic medicine, Amla, also known as the Indian gooseberry, Themblica officinalis.
I feel like Amla is everywhere in wellness right now. I see it in hair oil, in supplements, in juices. It's super high in vitamin C, right?
It is extremely high. It's one of the most potent natural sources of vitamin C on the planet. But for thermal aging, its role is all about structural defense. The research shows that Amla extract very potently inhibits MMP1.
MMP1. Is that the specific Pac-Man that eats type 1 collagen, the most important one?
That's the one. It's the primary collagenase. But Amla does something else really cool, something that none of the other ingredients do. It increases your skin's natural production of TIMP1.
TIMP1. Okay, let me see. Tissue inhibitor of metalloproteinases.
You got it. And the name tells you exactly what it does. If MMPs are the Pac-Men eating the collagen, TIMP1 is the police officer that shows up and handcuffs them.
Oh, wow. So Amla doesn't just stop the factory from producing more of the bad guys. It actively sends in the police to arrest the ones that are already running around causing trouble.
Yes. Amla boosts the police force that's naturally present in your skin.
I love that. So Amla handcuffs the Pac-Men, but didn't the notes also say something about it mimicking a growth factor? That sounds important.
It does. This is its third trick. It mimics the action of TGF-beta, transforming growth factor beta. This is one of the most crucial signaling proteins that tells your fibroblasts, your skin's builder cells, to make new collagen.
So Amla is a triple threat. It stops the breakdown of old collagen and handcuffs the destroyers, and it orders the construction of new collagen.
It is an absolute powerhouse. You can see why it has been used in India for thousands of years as a rejuvenating tonic. They understood that it worked even if they didn't know about MMP1 and TIMP1.
And speaking of construction, that leads us perfectly to the next member of the squad. Ginseng, Panax ginseng.
The classic adaptogen, the energy booster.
Yeah.
You usually drink this in a tea to stay awake during a long day.
But we aren't putting it in our coffee. We're putting it on our skin.
Right. And specifically, the active component that the research focused on is a type of saponin called ginsenoside RB3.
That is very specific. What does RB3 do that's so special?
It specifically promotes the synthesis of type I collagen.
Why type I specifically? Is there a type 2 I should worry about?
There are many types. There are over 20 types of collagen in your body, but type I is the main structural one in your skin. It makes up about 80 to 85% of your dermal collagen. It provides the tensile strength. It is what makes your skin tough and resilient and keeps it from tearing.
So if you lose type I collagen, your skin gets thin and fragile and tears easily.
Exactly. Ginseng ensures that the extracellular matrix, which is kind of the goo and the scaffolding that holds all our cells together, stays robust and functional.
So if the mushroom is the security guard, and apple mint is the interceptor, and Amla is the police force, then ginseng is the architect, coming in and actively rebuilding the walls, stronger than before.
That is a very effective way to visualize it. It really shows that anti-aging isn't one thing. You can't just solve it with one ingredient. It's a multi-front war, and you need specialists for each front.
There was one more herb mentioned in the research stack that had a really weird study attached to it. Comfrey leaves. Symphytum officinale.
Ah, yes, the zebrafish study. I was wondering when you were going to bring that up.
Please explain how we get from an anti-aging cream for humans to the fins on a zebrafish? Because that sounds like a bit of a leap.
Well, it sounds strange, but zebrafish are actually a fantastic model for biological research. They share a surprising amount of DNA with humans, their life cycle is very fast, and best of all, their embryos are transparent, so you can literally see what's happening inside them in real time.
Okay, so what do they do to these poor transparent fish?
In this particular study, they exposed zebrafish embryos to high levels of UV radiation. Now, biologically, UV radiation creates oxidative stress that is very similar to the stress from heat. It creates those same ROS, the same free radicals.
Wow.
They found that when the fish were in water containing comfrey extract, it protected their fins from damage. It dramatically reduced the cell death, and it preserved the physical structure of the fin.
It protected the fins from a sunburn, essentially.
That's a good way to put it. The compounds in the comfrey absorbed the UV and reduced the ROS production significantly. It basically acted as a biological shield. The implication is that on human skin, it would provide a similar barrier against environmental stress, whether that stress is coming from UV or from heat.
It is just amazing to think about all these connections. You have a mushroom from the Rocky Mountains, a mint plant from Europe, a gooseberry from India, a root used in traditional Chinese medicine, and a leaf that's tested on fish, all converging on this one very modern problem, keeping our skin from cooking itself.
And it really highlights a major shift in how we think about skincare and even medicine. For a long time, the solution to pain or inflammation was synthetic lidocaine to numb an area, hydrocortisone to suppress the immune system.
Right. Heavy hitters, drugs with side effects.
But here we are seeing these elegant biological solutions. The fungal extract doesn't just numb the whole nerve like lidocaine, which would leave your face feeling like you just went to the dentist.
Not a great feeling.
No. It interacts with the specific receptor mechanism. It's more targeted, it's more intelligent.
It's smart skincare versus brute force skincare.
And it forces us to redefine what sensitive skin even means. It's not just a feeling or a cosmetic issue, it's a biological vulnerability. If you have these overactive TRPV1 receptors, you are fundamentally more susceptible to thermal aging. Your skin is literally aging faster because it is panicking more often than someone else's.
That is a sobering thought. That stress, even cellular stress, ages you.
It does. And the synergistic effect is real. The heat from IR radiation sensitizes the skin, making it more vulnerable to UV damage. The UV damage then weakens the skin barrier, making it more vulnerable to pollution. It's a vicious cycle, and breaking that cycle at the very beginning, at the TRPV1 receptor, is a really strategic and powerful move.
So next time I'm sitting in the sun feeling that warmth on my face, or pressing my phone against my ear on a long call, I'm not just going to think, oh, this feels nice, or checking my emails. I'm going to be thinking, oh, my TRPV1 channels are flying open, calcium is rushing in, and the Pac-Man are waking up.
A little bit of healthy paranoia can be a good motivator, but mostly just remember that heat is a real factor, and that nature, in its complexity, has provided a surprising number of off switches for that damaging process.
Really makes you wonder what else is out there hiding in the dirt.
That's the most exciting part for me. We found this specific Grifolin molecule in this one mushroom, Albatrellus ovinus, but the forest floor is just teeming with fungi that we know almost nothing about.
Right, we barely catalog them.
Estimates are we've identified and studied maybe 5% of the world's fungi. Maybe. So if a humble Sheep Polypore can turn off a heat receptor, what can the other 95% do? Are there mushrooms that turn off the ISH receptor specifically? Or ones that trigger collagen growth directly without the need for ginseng or amla?
We are only just scratching the surface of the mycopharmacopoeia, the library of fungal medicines.
The chemical diversity in fungi is staggering, and we've analyzed such a tiny fraction of it. Every hike in the woods is a walk past a potential unread pharmaceutical library.
Well, I, for one, will never look at a drab, cracked, tan mushroom the same way again. It might just be the fountain of youth in disguise.
Or at least the fountain of not feeling the burn.
I will take it.
Me too.
So, to our listeners,
Yeah.
take a look at the ingredients list on your skincare. Look for Albatrellus ovinus. Look for Grifolin. And maybe, just maybe, use the speakerphone function on your next long conference call. Keep that hot phone a few millimeters away from your face. Your collagen will thank you for it.
That is sound advice.
Thanks for diving deep with us today. Stay curious, stay cool literally, and we will catch you on the next deep dive.
Goodbye, everyone.